It is my very great honor to introduce our fourth speaker, Claudia Mimoso. So Claudia did her undergrad at Cornell before moving to Harvard Medical School for her PhD with Karen Ehleman. And I've been fortunate to work alongside Claudia these past 4 years in the Ehleman lab. And I need to keep this short, but I'll just quickly note that Claudia really is an exceptional scientist and person. Scientifically, both in how she approaches and writes about questions, which are reflected in her NSF fellowship and in her experimental and computational skills. I mean, in our lab, she's the go-to person for ProSeq training and help, which really does say a lot. And she's also developed extensive lab analysis pipelines that we all use. She's put together an incredible body of work for her thesis, including the story she is presenting today, a project for which she has performed every single experiment and analysis. And all I say, I'm very excited to host Claudia for this talk today. So please join me in giving her a warm, albeit virtual welcome for her talk titled U1 snRNP increases RNA Pol II elongation rate to enable synthesis of long genes.
Thank you, Ben, so much for that very kind introduction. I'd like to thank the organizers for this opportunity today to share my thesis work, which focuses on the crosstalk between the transcription and splicing machineries. So the formation of a mature mRNA absolutely relies on RNA polymerase II transcribing processively from the transcription start site all the way to the gene end. However, as easy as this may sound, productive elongation is not a guarantee, and the polymerase must overcome a number of obstacles when transcribing long mammalian genes. These obstacles include nucleosomes, as well as sequence elements that can elicit stalling, arrest, or premature termination. In fact, anything that slows the polymerase down can be considered to favor termination, since nucleotide addition and termination are in kinetic competition with one another. Thus, the longer a gene is and the more obstacles the polymerase encounters, the more likely the polymerase is to terminate prematurely. And as such, a mature mRNA is not produced. Thus, to minimize premature termination, eukaryotic cells encode numerous elongation factors that either stimulate elongation or prevent stalling, arrest, and termination. These elongation factors are critical to prevent the formation of truncated and aberrant transcripts, particularly for long genes, which are enriched for several essential biological processes, such as DNA repair and neuronal development. As a result, abnormal expression or mutations in these elongation factors have been implicated in several diseases and have become an emerging therapeutic target. When thinking about gene lengths, of course, the largest contributor to increased gene lengths are introns. While introns are beneficial, for example, in increasing protein diversity through alternative splicing, they do come at a cost. A key feature of introns is that they are more AT-rich than exons. And since AT-rich sequences result in weaker DNA-RNA hybrids within the polymerase, thus lessening its stability, this increases the likelihood that the polymerase will either pause, arrest, or transcribe negative signals that cause premature termination. I want to highlight that premature termination of the polymerase within an intron typically results in the retention of intronic sequences within that released RNA. In several diseases, people have shown that inappropriate translation of these retained intronic sequences have been found to create neoantigens that impact cellular function and immune surveillance. Thus, overall, at a fundamental level, this means that long AT-rich introns, which have grown to become so pervasive within mammalian genes, really pose a major threat to transcription. So this begs the question, well, how does the polymerase contend with transcribing these long AT-rich introns? To answer this question, I used a repertoire of sequencing techniques to evaluate RNA synthesis, RNA Pol II density, and elongation rate, predominantly mouse embryonic stem cells. And the data that I'll be showing you today was recently published in Molecular Cell. So to evaluate RNA synthesis, I used a technique called transient transcriptome, or TTseq, which involves metabolic labeling of RNA during a short pulse of forest U, followed by selective enrichment and high throughput sequencing of newly synthesized RNAs. To monitor elongation properties, I took active protein coding genes and divided them into bins into 500 nucleotide bins that tiled all along the gene body, excluding bins that contained the start or the end site. This tiled bin approach allowed us to measure elongation properties at high resolution across mammalian genes. So first, I evaluated RNA synthesis within each bin. And in doing so, as depicted at this example gene, we find that TT-seq signal showed a gradual increase within the gene, in general agreement with reports that the polymerase accelerates as it transcribes across gene bodies. To evaluate RNA Pol II density, I used precision run-on sequencing, or PRO-seq for short, which allows for single nucleotide resolution mapping of engaged RNA Pol II complexes through the transcriptional incorporation of a single biotin NTP, which can then be used to isolate nascent RNAs. Typically in PRO-seq, we see the accumulation and drop-off of signal downstream of the TSS, and this corresponds to the promoter proximally paused polymerase and its subsequent release into productive elongation. This is a key feature of metazoan transcription. I want to highlight that higher PRO-seq density can reflect an increased residence time of the polymerase at each nucleotide position, which would reflect slower elongation. For example, slow elongation is typically observed around most transcript end sites. When focusing again now on RBM14, when looking at PRO-seq signal in red, we see that PRO-seq signal decreased sharply downstream of the promoter region, consistent with increased elongation rates as the polymerase transitions into productive elongation. However, we saw that PRO-seq signal then increased in locations within the gene body, particularly when the polymerase encountered an internal exon. This PRO-seq profile suggested to us that the high GC content of many promoter regions and exon sequences might reduce RNA Pol II elongation. So with these two data sets in hand, I was then able to approximate elongation rate, which I will be calling as the elongation index, as previously described by Patrick Kramer's lab, by effectively taking the ratio of RNA synthesis reflected by TT-seq divided by the number of polymerases as reflected by PRO-seq. And when now focusing on all active protein coding genes, where I took my bins and divided them into 4 groups based on the bin's GC content, and I then plot elongation index on the y-axis, what we observed was a generalizable trend where we observed faster elongation rates within bins of lower GC content, indicating that the polymerase would go faster in introns and slower in exons. To test this further, I then focused on the 3' splice site of internal exons and generated heat maps that are ranked by exon length with the largest exons towards the top. RNA-seq signal confirms the accurate definition of intron-exon boundaries, and I also observed elevated GC content over the length of the exon. I then probed for RNA Pol II using 2 orthogonal approaches, PRO-seq and RNA Pol II ChIP-seq, and both assays report a significant increase in polymerase density within the GC-rich exon as compared to the intron, indicating that the polymerase slows down when transcribing exons and then re-speeds up. Furthermore, this data illustrates that even short internal exons, as reflected on the lower half of this heat map, can elicit slower elongation rates. To summarize what I've told you so far is that we find that elongation rate is highly sensitive to changes in GC content, and that the polymerase is faster in AT-rich introns compared to exons. This increase in elongation rate within AT-rich introns by default reduces the likelihood of termination or arrest because of kinetic competition. But how does this happen? We next asked whether the splicing machinery might play a role for these increased elongation rates within introns, since many groups have reported crosstalk between the complexes that process introns and transcription. More specifically, recognition of the 5' splice site by U1 snRNP, which is the first step in splicing, occurs shortly after the 5' splice site appears in the nascent RNA. And U1 has been documented to enhance gene activity in a manner that influences a number of biological processes, altogether raising considerable interest in understanding how U1 accomplishes stimulating gene expression. So to determine if U1 could contribute to the acceleration that we were seeing in introns, I functionally depleted U1 by treating mouse embryonic stem cells with an antisense morpholino, or AMO for short, which is complementary to the U1 snRNA. This is a widely used approach to outcompete U1's binding to 5' splice sites. And after 4 hours of treatment, I then generated both TT-seq and ProSeq libraries. So first, I used my TT-seq to define differentially expressed genes. And in doing so, I found that 60% of active protein coding genes, shown in red, show reduced RNA levels following U1 ammo treatment. In agreement with work from the Dreyfus lab, we do find that downregulated genes are typically longer and have more introns than those unaffected. To assess whether this broad downregulation was associated with splicing, I separated out intronless transcripts, shown here on the right, and find that these RNAs are largely unaffected, with no biases towards gene downregulation. With the ProSeq, I was able to push this a bit farther, and I was able to identify downregulated genes that showed no significant reduction in promoter ProSeq levels, but instead exhibited a loss of gene body ProSeq, indicative of defects during productive elongation. This subset of genes will be referred to as elongation-regulated genes, and I just want to highlight that they are enriched again for long genes and pathways such as DNA repair. So to investigate elongation defects, again, I took my genes and I divided them into 500 nucleotide bins that tiled all along the gene body. I then summed ProSeq signal within each bin, and as expected, we see a very significant reduction in ProSeq signal at our elongation-regulated genes. This tells us the reduction we saw by TT-seq is really occurring at the level of transcription. To dig deeper into the mechanism, I wanted to compare this gene set to a set of genes that were unchanged by TT-seq and similarly showed no significant reduction in promoter ProSeq levels. You might predict that these genes would also be unchanged by ProSeq. However, they actually showed a significant increase in signal, which is intriguing because if you were making the same amount of RNA with even more polymerases on that gene, that suggests that the polymerase is moving more slowly. As such, I then calculated elongation index per bin, and in doing so, we find a broad and significant decrease in elongation indices, indicating that at even largely unaffected genes, the polymerase moves more slowly without U1. To evaluate this farther and probe the role of sequence content into this phenomenon, I compared elongation index, which will be shown on the y-axis, with respect to GC content on the x-axis in both scrambled and U1 ammo conditions. And this will be focused on genes that were unchanged after U1 inhibition to avoid altered read counts from impacting this result. So first, under control conditions, we observe faster elongation indices within regions of higher AT content. However, after U1 inhibition, this increase in elongation rates within AT-rich sequences was reduced, indicating that the polymerase is unable to achieve these higher elongation rates in AT-rich regions without U1 snRNP. So to build onto the model I've told you so far is that RNA Pol II acceleration in AT-rich sequences requires U1 snRNP. Without U1 to stimulate elongation rate, long genes are particularly sensitive to gene downregulation after U1 inhibition. So having established that inhibition of U1 causes slower elongation across AT-rich sequences, we next wondered, well, how does this result in the decreased expression of our elongation-regulated genes? To answer this, I first used a hidden Markov model to define sites at which the wave of ProSeq signal representing elongating polymerase drops off in U1 ammo-treated cells. And I was able to identify such transition points before the end site at about 48% of our elongation-regulated genes. This included genes that were previously reported by Phil Sharp's lab to undergo premature cleavage and polyadenylation in mouse embryonic stem cells. To globally validate my identified transition points, I made heat maps of ProSeq signal in both scrambled and U1 ammo conditions, and these heat maps are ranked by increasing distance from the start of transcription to the transition point, with the largest distances towards the bottom. These heat maps confirmed that we see a pronounced drop in ProSeq signal at our transition points only under U1 ammo-treated conditions, whereas we observe continued synthesis in control cells. Additionally, these heat maps demonstrate that our transition points are observed at variable distances from the TSS, with a median distance of about 2.8 kb. I want to highlight that the 5' splice site at these genes is at a median distance of 140 nucleotides from the TSS, indicating that U1 would be fully loaded before the polymerase reached this point in control cells. So now that I had these transition points, I next wanted to understand for how many of them can the loss of pro-seq signal following inhibition was a direct consequence of premature termination, given that previous work from the Dreyfus, Sharp, and Jensen labs has demonstrated that base pairing of U1 to 5' splice sites can somehow prevent premature termination. Very briefly, premature termination involves the recognition of polyadenylation signals, or passes, by the cleavage and polyadenylation machinery, or CPA. This leads to the cleavage of the nascent RNA and its subsequent polyadenylation and premature termination of Pol II. As a result, we can identify polyadenylated RNAs in our dataset to monitor CPA activity. And by mapping the 3' end of these RNAs before the poly A tail, we can track where cleavage happened. This can be done using a technique called poly A click-seq, or PAC-seq for short. Very briefly, this technique involves reverse transcription using an oligo-GT primer that will anneal to the poly A tail, and also includes the presence of chain terminating VTPs that will terminate the RT reaction shortly after the poly A tail is made. So to identify transition points that were associated with premature cleavage and polyadenylation, I generated PAC-seq libraries after U1 inhibition to monitor polyadenylated RNAs. I want to highlight that this was done using Forest U labeled RNA to focus on newly synthesized RNAs. I then searched for pass motifs that were found between the TSS and my transition point, and I focused on those that were associated with PAC-seq reads in U1 ammo-treated cells, meaning that these motifs were recognized and used by the CPA machinery. Furthermore, this analysis revealed that 47% of our genes with a transition point harbored a pass motif with detectable PAC-seq reads, or an actionable pass. This result was confirmed using an independent published dataset from Phil Sharp's lab that also identified polyadenylated RNAs after U1 inhibition. I found a similar percentage of genes with actionable pass usage and a strong overlap between our two datasets. I want to also highlight that I looked at PAC-seq signal in U1 ammo-treated cells at our actionable passes, and this metagene plot shows that PAC-seq reads were focused just downstream of the pass motif at genes that showed apparent premature cleavage and polyadenylation. Furthermore, I also evaluated actionable pass usage in control in U1 ammo cells by comparing the number of PAC-seq reads between the pass motif to +100. This further demonstrated that PAC-seq signal increased significantly near actionable passes after U1 inhibition as compared to control. And TCA1 is an example of this behavior, where PAC-seq reads are enriched just downstream of a pass motif after U1 ammo treatment. So to briefly summarize, we consider that this subset of elongation-regulated genes, how they can be explained, is that U1 inhibition leads to premature cleavage and polyadenylation. However, for more than half of our transition points, they fail to show evidence of pCPA, indicating that premature termination alone cannot explain all of the elongation defects observed in U1 ammo-treated cells. So what else could be happening? To investigate other elongation defects elicited by loss of U1, I focused on my genes that had a transition point, but without evidence of premature termination. And I searched for motifs that were enriched around these transition points as compared to mock sites selected at random at elongation-regulated genes that fail to have an identified transition point. I want to highlight that the distribution of distances between the TSS and my mock sites matched those of my target gene list to allow for correct GC normalization. So after running a few different motif algorithms, what we found is that motifs at these transition points were enriched for sequences that contain T stretches and a polypurine to polypyrimidine pattern, both of which resembled previously defined sites of backtracking and transcriptional arrest. Notably, an arrest motif was found at about 91% of our transition points without premature termination, further suggesting that these sites can reflect instances of backtracking and arrest of the polymerase. To probe this further, I then generated metagene plots of pro-seq signal aligned to the arrest site that was closest to our transition point. And I'm going to be showing you the difference in prosecretes between U1 and scrambled ammo conditions. This plot revealed a significant accumulation of signal upstream and a significant reduction in proseq signal downstream of our arrest motif, further demonstrating that this is consistent with the increased occurrence of backtracking and arrest of the polymerase at these sites after U1 inhibition, as observed at MFF and FLIRT3. As such, we conclude that the absence of U1 renders the elongation complex more prone to transcriptional arrest, consistent with reduced elongation rates, resulting in multiple defects in RNA synthesis. So to leave you with one final model of the work I told you today, to promote full-length synthesis of mammalian genes, RNA Pol II elongates faster in AT-rich introns compared to GC-rich exons in a manner that's dependent on U1. Without U1 to stimulate elongation rate, the slowed elongation complex is then more susceptible to both termination and arrest. Long genes are particularly sensitive because the extended gene body presents more chances for termination and arrest to occur. Our work answers long-standing questions in the field regarding how the polymerase contends with transcribing these long AT-rich introns, and also provides mechanistic understanding for how U1 influences gene expression. Given our work and recent structural work by Patrick Kramer's lab, showing a direct interaction between the polymerase and U1, we propose that U1 functions as an elongation stimulatory factor. Lastly, our data raises another question. If the main goal is for the polymerase to just reach the gene end, why wouldn't Pol II just go faster everywhere? Why would we see a sequence-dependent effect? It's important to note that increased elongation rates come at the expense of reduced fidelity. Our work would suggest that RNA Pol II fidelity is lower in introns compared to exons. We hypothesize that such transcriptional errors are more readily tolerated in introns, as these sequences are excised during splicing, whereas exons require high fidelity for translation. Thus, we propose that cells use U1 to stimulate elongation in introns, where the benefit of protecting the polymerase from termination or arrest completely outweighs the risk of introducing errors during transcription. And with that, I'd like to thank everyone in the Edelman Lab, especially Karen, for all of her mentorship and support over the course of my PhD, the Nascent Transcriptomics Corps for always being willing to talk about sequencing techniques, my thesis committee, funding, and you all for your attention. I'm happy to answer any questions.
Fantastic talk, Claudia. That was great. So a few questions already. And also remind people that you can either click the raise hand button or type your question into the Q&A box and I'll read it out. So Sergey has a couple questions. I'll start with the first, which is, do you see changes in Pol II elongation at downstream splicing sites?
Downstream splicing sites. So yeah, so the changes that we see in elongation with corresponding to splicing is really at first very evident. So kind of in the heat maps that I showed, where you see the slowdown of the polymerase when it's transcribing the exon, it's more so the polymerase responding to that GC content than it recognizing the actual splice site itself. But the splicing machinery is then helpful, you know, as the polymerase is transcribing based on sequence content.
I think you largely answered the second question in your finishing up slide. But do you expect more transcription errors at faster elongating regions, including downstream exons?
Yeah, so that's something we're really excited looking at is transcriptional errors with respect to this work.
Yeah, and then Alan Underhill says, first, great talk. Second, do you see any relationship with repeat sequence types, frequency, or distributions in terms of elongation rates?
Wow, repeat sequences. My work is largely focused on just like pure GC versus AT content, but that is something for us to consider looking into further. I haven't looked at it yet. But.
And then Jason Fan says, hi, very cool talk. I was wondering whether U1 also helps with elongation on the exons and whether its association with Pol II is altered between introns and exons. Yeah,
great. So yeah, that is something I think still we need to further understand. So it could be for some, but maybe not for all. So splicing of the intron occurs very shortly after the intron is made, so in that downstream exon. And what Patrick Kramer's lab has shown structurally is that as you keep going and splicing, it completely clashes with the U1 Pol II structure. So you can only get a few steps in there. So if it kind of depends on the rate of splicing for how much U1 could potentially help in that region, if that answers the question.
Yeah, and after it's done like splicing, does it stay associated with Pol II or does it come off?
Great question. I think we still have a lot to learn. We don't know.
And then there was a question in our organizers chat that I don't know the answer to, which was, is something, do you think U1 is this conserved all the way to yeast, which most genes don't have introns, but does U1 associate with Pol II genes in yeast to stimulate elongation?
So actually, so the structural paper did look at this, and the regions that for the interaction between U1 and the polymerase are conserved across metazoans, but not really in yeast. And for potentially that exact reason that in yeast, they either have no introns or they're so tiny, so they don't have as big of this problem that introns can bring. So it is, I think, really cool how this may have developed for metazoans as introns have really just expanded to really just make sure the polymerase gets to the end. But yeah, great question.
And have you thought about looking at like how like this system has evolved? Like some of the key bits are conserved across metazoa, but are there just like important differences between how this works in humans, which have very, very long genes and introns, versus something like flies, which are relatively shorter?
Yeah, that's something I've been thinking about, as well as like the regulation, the overall regulation of the SNRPs. I think we still have a lot to learn to understand that complexity as well, because U1's also been implicated in several developmental systems to regulate isoform switching. So I don't really have a full answer to your question, but I think that is something to consider across, flies and humans.
And then maybe one last question. There's a few that we haven't got to. I missed there were a bunch more of the Q&A. I scrolled down and saw all these questions. But one last question from Nicole, who says, great talk. You mentioned neuronal genes are long. Do you think that this faster transcription plus lower fidelity may be applicable to DNA damage associated activity-dependent transcription observed in neurons?
Maybe. I would need to read up a little bit more on that, but that's a really cool idea.
Great. And then we'll finish up there, but I'll note there are a few other questions. And so you just type in your answers for the people who ask them. And thank you again, Claudia. I'll pass the moderation over to Skye.
Thanks, Ben.
Great, Claudia, thank you for the wonderful presentation.